is a repeating thing. It goes up, it turns around, and comes back down, and it just goes like this over and over. And so what you get is a sheet of amino acids versus a coil. Then there's the tertiary structure, which is the third level of structure. And so we have sheets, we have coils, but then it's how they're oriented to each other. Because sometimes you don't all just have coils and sheets, but you have these linker regions that connect sheets to sheets in coils. And so how it looks like this is the tertiary structure. It's how one strand folds on itself. And then there's finally quaternary structure. If you take multiple strands and put them next to each other, it's how two strands or three strands will connect to each other, and how they'll orient to each other. Hemoglobin is a good example of that. There are 4 strands that go into one hemoglobin protein. And so you have 1, 2, 3, 4. So proteins have a number of different functions. Because we've talked about enzymes. Enzymes have a purpose, a functional purpose that they change things in our cells. Proteins also serve structural functions. Keratin is very, very present in our skin, in our nails. We'll talk more about this in the tissue section. But keratin is just a tough protein. It's for mostly protection. And then there's collagen. Collagen is also for structural purposes, but it's down in our skin. Keratin is on the surface, it's very hard. Collagen is deeper down in our skin. Protein is also used for communication within our body. Our body can put proteins into our bloodstream, which will tell other cells in our body that something is going on. Some hormones are proteins. Ligand is a term for something, another molecule, that will stick to a protein. So you have a protein, and then there's a ligand that will stick to it. Something like hemoglobin or oxygen is essentially a ligand that's going to come in and bind to that protein. A receptor is a protein that is going to bind a ligand for the purpose of sending a signal. It sits there waiting for a ligand to come along, and when it sees it, it sends a signal. Imagine something like an estrogen receptor. If there's estrogen in our body, it's floating around, it's going to bind to a receptor that is made to recognize estrogen. And estrogen is a hormone. And so when that hormone is recognized, a signal has to be sent to the cell that recognized it. So the receptor will bind estrogen. Estrogen is the ligand, and so it will send a signal to the cell. Some proteins are for transporting things across the cell membrane. A cell membrane is pretty solid. Bigger molecules can't get across that membrane. Glucose can't just go into a cell. Some proteins will act as a pump molecules into a cell. Some proteins will act as a hole, so it'll make a break in the cell membrane that allows things to pass through. A carrier is a protein that will take a solute, something like sodium or potassium, from one side to the other. A channel protein is essentially a hole that will decide what can and can't go through it. So it doesn't actively take something from one side to the other, but it will decide what can go through it. And then there's our enzymes. Enzymes are catalysts. We said an enzyme makes some reaction happen. Most of these enzymes are globular proteins. A globular protein is going to be circular or spherical, whereas things like membrane transport proteins are going to be longer and skinnier to get through the cell membrane. We talked about glycoproteins. We said that's for signaling. It's like having a flag on the outside of the cell saying, I am a liver cell, or I am a kidney cell, or I am an immune cell. Antibodies in our bloodstream that recognize things that are invading our bodies, bacteria and viruses, are proteins. You take different proteins in different sequences, put them together to recognize different things in our bloodstream. Our muscles are primarily protein, right? If you want to eat more protein in your diet, you're going to eat meat. And when we eat meat, we generally are eating muscle. Muscle is very high in protein. Those proteins are used to change shape, which allows us to move our body parts. Proteins are also used for cell adhesion. Cells need to stick to each other. If they didn't, you would just fall apart. And so proteins can act as glue to hold each cell together. So you get two cells with proteins sticking out. When they come together, the two proteins touch each other and they stick. And now those cells are stuck together. We're going to talk more about the enzymes because the enzymes are really, really important. So we said the enzymes are proteins that are catalysts. They allow reactions to happen at body temperature. The reactions that happen in our body would happen regardless, but you generally have to heat them up to make them happen. But we are 98.6 degrees. If they're not going to happen spontaneously at 98.6 degrees, we need a catalyst to make them happen. So an enzyme is going to bind something that is going to change. An enzyme grabs something, and then it changes it, and then lets it go. The thing that it's going to grab is called the substrate. That could be a sugar, it could be an ion, it could be another protein, it could be anything. But whatever that enzyme grabs and changes is going to be our substrate. When we name an enzyme, we take the substrate and then we add ASE on the end. And so amylase is the enzyme that digests starch. Another name for starch is amylose. And so if you break down amylose, you're going to do it with amylase. We drink lactose in our milk. You have to break it down with lactase. People who are lactose intolerant have lower levels of lactase. In general, an adult human, if you're not drinking milk, will actually lower the amounts of lactase in our body. And so by continuously drinking milk, we keep the lactase levels up, so then we can continue to digest lactose. People who are lactose intolerant, even though they're drinking it, have a genetic coding in their genes that tell them to shut that off. They don't have as much as the rest of us. We talked about how enzymes lower that activation energy. Remember, for a reaction to happen, you have to get over that hump. If you can lower that hump, it's much easier to happen. Got a little video here. This is just going to show enzyme work. So here's our enzyme, and it's shaped to have this perfect little cleft for a substrate to come into. That cleft is called the active site. That's the part of the enzyme that actually functions. So now we're going to have a couple of substrates come in. They're going to fit together, and then once they're together, they're going to bind into their parts on the enzyme. Now the enzyme is going to act on these substrates. It's going to combine those two substrates together. So now we took two parts and combined them into one. And once that happens, the enzyme is going to release it, and then the product is just going to flow away. Once that happens, the enzyme will revert back to its shape, original shape, and it can act again. An enzyme can do this over and over and over and over. It's not a one and done thing. A catalyst can be used over and over. This is essentially what we just looked at, so I'm not going to spend too much time on it. But remember, we have our enzyme with pockets that our substrate will fit into, and then the enzyme will act on that substrate to do something to it. Here, we took a disaccharide and we're going to break it apart into 2 monosaccharides. So this is sucrose, and so the enzyme is sucrase. We said that enzymes can be used over and over again. They're very, very fast. That breaking apart of a sucrose molecule happens incredibly fast. And so as soon as one is done, it'll go to another and another and another and another. We've talked about how temperature, pH, will change that shape. So we don't need to talk about that. A lot of enzymes on their own can't do their job. They need what's called a cofactor. And so it says 2/3 of our enzymes require this cofactor. And a cofactor is something that's going to bind to that protein, and that cofactor is not another protein. When it binds, a lot of times what it's going to do is it's going to cause the protein to change shape. So if we go back and look at this, right now it has the correct shape to fit that substrate. Imagine we have a different enzyme that does not have the correct shape. But once you take a cofactor and put it on the protein, it changes shape, and now it will fit the substrate. A coenzyme is an organic cofactor. And usually these are water-soluble vitamins. So things that you take in your multivitamin, niacin and riboflavin, these are coenzymes. You might have heard of coenzyme Q10, something that people take as dietary supplement. That is a cofactor that's part of the metabolic process in your mitochondria breaking down sugar and making energy. Without that coenzyme Q10, you break the chain. So the enzyme does its job, and it made its energy, and it's going to pass that energy on, but there's nothing to pass it to. So the coenzyme is something that sits there, accepts the energy, and then is going to pass it on to the next enzyme. Another thing that's involved in that metabolic process is something called NAD. So the metabolic pathway, like I just was mentioning, is not one step. It's one step, and then another, and another, and another. And so let's say we have something like glucose. We're going to say glucose is A. In the end, we need to get to D to get our energy out of it. We can't go straight from A to D. So one enzyme alpha, is going to turn A into B. And then beta will turn B into C, and then gamma will turn C into D. Almost all the processes in our body are chains like this. They're pathways, which is a good thing. That means that if one enzyme here goes haywire and becomes overactive, the whole thing doesn't become overactive. If B decides that it's going to double its productivity, it's not going to make a difference in how much D we make, because B can only make as much C as A turns into B. Does that make sense? I think I mixed up a B and a beta. Beta is the enzyme. If beta becomes overactive, it can only act on as much B as alpha made. So it's redundancy. Cancer in our body breaks redundancy. It overcomes that, and it says, well, I'm going to skip this. I'm going to find a way to go straight from A to D with enzyme zeta or something like that. So it will skip all of these safety processes. Our last type of organic molecule we need to talk about are nucleotides and nucleic acids. This includes ATP, which we said was the energy currency. A nucleotide has 3 parts. There's a nitrogen base, there's a sugar, and then there's a phosphate group. Make sure you know these 3 parts of a nucleotide. The best known nucleotide is that ATP. It is a nucleotide. It goes into making DNA and things like that, but it's also that energy currency. And it's made up of three parts: adenine, which is the nitrogen base, ribose, which is a sugar, it has the OSE at the end, and then there's a phosphate group. It's ATP, this is adenine triphosphate. And so there are three phosphates. ADP is adenine diphosphate. So there are only two phosphates. So this is ATP. Okay? So this is adenine. This is what a nitrogenous base looks like. There's a bunch of nitrogen in it. And then there's a sugar down here. And then there are phosphates. Okay, here this goes ATP. There are 1, 2, 3 phosphates. A phosphate is a phosphorus with some oxygens around it. Down here, this is adenosine monophosphate. It's the same here, it's just there's only one phosphate instead of 3. So this would be AMP. ATP, ADP, AMP. It's important for energy. We've talked about those things. When we need to break apart ATP, get the energy out, we use an enzyme, ATPase. The enzyme that works on ATP, ATPase, is going to break apart those phosphates. And so it's going to break off a phosphate on the end, we get some energy. We break off another one, we'll get more energy. Phosphorylation is the process of putting a phosphate group onto something, usually a protein or a carbohydrate. If you take a phosphate and you put it onto something, it's going to be called phosphorylation, and the enzymes that do that are called kinases. Kinases take phosphates from one molecule and transfer them to another. So here's an example. We got glucose, and when we break down glucose in our body, we use oxygen. So we're going to take glucose, combine it with oxygen, and we're going to make carbon dioxide and water. When we do that, we're going to release energy. And when that energy gets released, it's going to get captured by ADP. ADP is low energy. The energy from this breakdown up here is going to get converted into a bond holding an extra phosphate, an extra P, onto ADP to make ATP. The ATP can then go wherever it needs to go in the body, whether it's a cell in our muscles or our brain or anything like that, and then it can be broken back down so it can release that energy. So we can make energy in one part of the body, transfer it to another part of the body, and release it there for whatever is needed with that cell. So this is actually a two-step process. If we take glucose, and we break it down, that's called glycolysis. So breaking down of glucose is glycolysis. We make 2 ATPs by doing this. We turn glucose into something called pyruvic acid. If there's no oxygen available, that's the end of the line. We can't break it down anymore to get more energy, and so our body converts it from pyruvic acid to lactic acid, and then our body gets rid of it. And we've gotten 2 ATPs out of it. Although most of the time we have oxygen available. And so we go through what's called aerobic respiration. Pyruvic acid and oxygen go into the mitochondria, which we'll talk about next week. It's part of the cell used for making ATP. And then it will go through a longer process that will get us 36 more ATPs out of it. So in the absence of oxygen, you make lactic acid in 2 ATPs, in what's called anaerobic fermentation. When we have oxygen, we do aerobic respiration, and we get an additional 36. And so why on earth would our body ever do this if we get so much more energy out of this?
Waste.
What about waste?
The lactic acid.
Lactic acid is a waste, yes. And so we actually don't want to make lactic acid. And so there's a down, not only do we not make enough ATP here, but we make a waste product that we don't want. So why would we do this? Yeah.
We can only take in so much oxygen.
We can only take in so much oxygen. If you exercise really hard, you can get cramps, right? It's a buildup of lactic acid in your muscle. The oxygen gets used up in your muscles. We have so much oxygen in our muscles, so much in the blood, in our bloodstream, but we can use it up a lot faster than we can take it in. And so our body's going to run out of oxygen, but it still needs energy. I mean, if we have no more energy, we die. And certainly if we're going to keep running, we need more energy. And so our body will do this. It'll say, well, 2 ATPs is better than no ATPs. Then we do this. But as soon as you stop running and you catch up, you're going to shift back to the aerobic respiration. You can also take nucleotides and string them end to end to make polymers. Again, a string of nucleotides in a polymer is called a nucleic acid. We have a couple different types of nucleic acids. Number one, the most famous, is certainly DNA, right? Deoxyribonucleic acid. There are millions of them, strung end to end. It's a giant polymer. This is what makes up our genes, and these are the instructions for making the proteins. Proteins do not replicate themselves. Proteins come from genes. Genes are the instructions for making those proteins. There's also RNA, ribonucleic acids. So these have one more oxygen than these, so deoxy, and these are just ribonucleic acids. And there are 3 types of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. We'll talk about the specific purposes of those next week. These are big, 70 to 10,000 nucleotides, but they are not as big as DNA. DNA contains a whole bunch of genes. Each RNA comes from one gene. We'll see the process of making a protein, but we go from DNA, we use DNA to make RNA, and then RNA is used to make proteins. Okay. Whew. Is everybody still alive? Again, I don't know how to make that more